Talking with Greg Tylka here at Iowa State. He's got done with your presentation on soybean cyst nematode. First of all, thanks for hosting such an informative event here like we had.
Yeah, Dustin, it's fun to do because you get to see other people's excitement about soybean cyst nematode. I got to be careful not to act too excited about a terrible pest, but my job is to help farmers battle that pest. And so it's fun to hear the questions and know that we're working on the right things in terms of our research.
Okay, so we usually talk a 30,000-foot view, but you really got more in depth here. And some of the things I found interesting was we were talking about the anatomy of the nematode to understand how it works. People always think of a bug. They don't think of it as being a worm.
Yeah.
So tell us a little about that and maybe just give us a quick example of what it kind of looks like.
Sure. So it's microscopic. The worm is about 1/100th of an inch long. And it's confusing to people perhaps also because it goes into the root and it attaches to the vascular tissue of the soybean root, but then it swells up. The adult female eventually gets so big that she pops out of the root, and you can see her as a little white dot on the surface of the root. And in fact, that's one way we advise that farmers and agronomists check fields for SCN. Wait till 5 or 6 weeks after planting, and then pretty much through to August, maybe mid-August, dig roots and look for those little white females. You can see, even though the nematode's a microscopic worm, you can see the swollen adult females with your naked eye. They're about the size of a period at the end of a printed sentence. So a newspaper article or a textbook, the period at the end, that's about as big, but it'll be white colored and about that size. And you can see them with your naked eye.
And then the males kind of go back to the worm stage, revert back and kind of do whatever they've got to do. And then what was interesting was the breeding process. They're not a monogamous pair like we see it in many different species around the world. Explain maybe how that reproduction cycle works.
Yeah, and it creates a problem. So it's simple. There's, like humans, males and females that have to mate for offspring to be produced. The female produces all the offspring in the form of eggs. But the interesting and problematic part is a male will mate several different females, and a female will be mated by several different males. So the eggs, if a female makes a total of 250 eggs, they might have 200 different genetic types because of the intermating that occurs. So they're not clonal. Clonal would mean that all the eggs are just like the mother's genetics. They are a mix of genetics of all the different males that mated with the female to produce those eggs. And that genetic diversity allows the nematode to adapt. And that's why we've seen the nematode adapt to and overcome the common PI 88788 resistance because of that genetic diversity.
And that's where I was going to kind of go next, and we'll kind of circle it back around. But the PI 88788, we were talking about that last week with some folks at BASF, and they're talking about it's not completely useless, but it's really been beaten into the ground, and the nematodes have adapted. We're seeing the Peking rise, and they're really starting to worry that it's going to get hammered just as hard. And you kind of gave us a warning about Peking versus PI 78. Maybe tell our listeners about that.
Yeah, so we believe, and we've seen a little bit of it, that the Peking resistance, well, let me back up. 88788 resistance took about 20 years to start to fall apart. And here we are 30 years into it, so it's really falling apart in that there's lots of nematode reproduction. Peking, we don't believe, is going to be as durable. And it's kind of counterintuitive, but it's not going to be as durable because it's more effective. So 88788 always allowed a little bit of reproduction. And so the nematode population in a field over decades always had a vulnerable part of its population. Peking is so effective that the tiny, tiny percentage that overcomes Peking, it's truly able to reproduce, and over time it's going to build up quicker than the slow curve that we saw for 88788. So Peking works great at first, and we're not seeing it fail by any means, but we're a little nervous because by nature of how quickly it works and how effective it works, it's going to be vulnerable to breakdown quicker than 88788. So the good news is we have hundreds of 88788 varieties out there still, and we need seed companies to don't abandon those. In my dream world, I wake up some morning and find that there's 400 varieties with Peking and 400 varieties with 88788, and farmers grow one year, 2 years later, they grow the other.
So obviously, we're thinking about Peking. I was just starting to think about it as you were explaining. Obviously, it wouldn't be a nuclear option then, because some people might think, well, if it's that effective,
Yep.
why don't we just hit it hard
Yep.
and get rid of them?
Yep.
But there's still that microscopic chance that
Yep.
they're going to get through it.
Yep. Yeah, and my money would be on that microscopic chance. So it's going to look like a nuclear, like you're nuking it. We see 20 bushel per acre yield responses by going from worn out 88788 resistance to Peking. But don't fool yourself. Mother Nature finds a way. And that Peking is not immune. It's extremely resistant, but there's a small part of that population that is going to build up quicker than 88788, which had a larger population of the nematode able to reproduce, but that actually slowed it down over the course of decades. So we desperately need people to use both, alternate between the Peking resistance and the 88788 resistance.
Now, I know you're on the, obviously the university side of it, not the commercial side, but we had the folks at BASF here helping as well. We talked to them last week in Kansas City at our convention, and they brought up the fact that they're trying to bring tools out here, hopefully by 27, 28, that'll at least help boost things like PI80. I mean, are we going to be needing to rely on that? Because the alternation obviously is going to help a little bit, but if it's that, are we going to still have to require on those helping hands now to help get those two to keep being effective?
No, I think the new BT trait, which I believe they're going to call Nemosphere when it's rolled out, is going to be an incremental improvement. But I don't think if we alternate regular 88788 and Peking, we're going to see improvement on 88788 and its yields by itself. And then I think the new Nemosphere trait is going to be in an 88788 background. So I'm at this point thinking of it as it'll be new and improved 88788, but it still probably should be alternated with Peking. And I just think growing Peking alternating with regular 88788 is going to improve 88788 performance.
And in the beginning, you showed us how the rate of increases. These products, these seed treatments are going to help delay that first generation getting going. Thing is, we learned the eggs can stay dormant for up to 10 years. It may just prolong how long it takes them to get going. But the average cycle, we can get 5 or 6 generations a year. You're saying if we start with you made you pulled out an arbitrary number of 200 eggs. If you started the season with that, you'd have 40,000 by the end of the year.
Yeah, it's going to be a lifelong battle. And we all farm in Mother Nature's world. It's not the other way around. So it's going to be a battle, but resistance, the nice thing about resistance is it's in every single cell in those plants, every single root cell above ground. But we need also to take advantage of other strategies like the seed treatments that might protect the roots only for a month or two, but that's added protection. And then also alternating with corn, because there are a certain number of eggs, not enough, but a number of eggs that'll hatch in a year of corn, and those are guaranteed to die off as well. So we need to use all those things in an integrated approach that we used to call integrated pest management. We need to use all those tools. None of them is a cure in and of itself.
And what I found really interesting is the numbers that we see come out of a sample. We were talking about that, and we'll have that video. Talking about they could be into the thousands per acre. But you're talking about an animal that has a 99% death rate when it's out in their nature. I mean, so how does that, what are we looking at here?
It's mind-boggling, and that's right. I presented some information that 99% of the eggs that are produced by a female probably aren't going to live on to produce new females. But it just shows you the astronomical power of numbers in that even though it's that 1%, over time, 3 or 4 or 5 turns of the life cycle, it can really build up and get away from us. And that's why farmers and agronomists need to be proactive, check fields for SCN, and then really take it seriously and start using all those tools, different types of resistance, seed treatments, non-host crops.
A couple more questions I have for you. Going back now to the physiology of these insects, or bugs, or whatever you want to call them, pests.
Yeah.
They inject with that little stylus, similar to what I would imagine like the diabetes testing, a little needle. They inject something into the plant and then to make that plant alter its genetics or however its makeup to make them be able to feed. Explain to me a quick 30,000-foot view of how that works.
Yeah, it's a fascinating biological system. That nematode does have a hypodermic needle-like structure. It injects chemicals into living plant cells, and it changes their biology. And it develops into a feeding site for the nematode. And that feeding site, those cells that feed the nematode, are unlike any other cell in that plant. They don't look like a normal plant cell. And furthermore, what's really cool biologically is that if we reached into the root carefully and killed with a needle the nematode, that feeding site withers and dies away. It doesn't revert back to a normal plant cell, it dies away. So the nematode hijacks the biology of plant cells to make a food factory. And that's the holy grail of science for us. We have molecular nematologists and plant pathologists studying what is that chemical in the nematode that is injected through the stylet into a living plant cell that completely changes the biology. And we've made great strides, but I honestly can't tell you, they can't tell you if we'll have the answer in 2 years or 5 years or 10 years. But it's interesting to think about how this all evolved, and Mother Nature is just something to behold. The complication of it all. We've worked through all the simple answers, and could it be this chemical or that chemical? None of those came to be. But we're still making incremental increases in knowledge, and I have confidence someday we'll figure out how the nematode is making that feeding site, and that's the step towards preventing that to happen. We can genetically engineer making that not happen once we know how it happens.
So obviously, you kind of talk about the beauty of Mother Nature. It's got to be kind of an exciting rock and a hard place kind of thing, or juxtaposition. The fact that you want to prevent these things from happening, but yet sit there as a biologist and look at the awesomeness of what nature can do. It's got to be impressive at the same time.
Yeah, it is a bit of a paradox, but at the end of the day, I love working for farmers, and that's what Iowa State wants me to do. That's what they pay me to do, and I get to experience lots of amazing biology with soybean cyst nematode while doing that. But I can tell you, if in magically 6 months from now, I have a discovery that would completely eliminate soybean cyst nematode, I'd pull the trigger on it, and I'd have something else to study. So my heart and my interest is first in the farmers, and they've wonderfully supported our research, and I get to experience lots of biology, but I'm always working towards getting rid of those critters. I sometimes seem too excited about the biology of the nematode, but if I could snap my fingers and have them all disappear, I'd do it in a heartbeat.
I think we'd expect nothing less, but it's still got to be awesome as in your field to watch something like that happen. It gives you a better understanding for other things as well.
No, it does. And I remember I'm about to speak at the ISU Integrated Crop Management Conference in Altoona next month. Last year I got talking about it, and I mentioned that it's so cool to see things that I was taught from a textbook as an undergrad play out in a farmer's field. But I got to be careful when I say that, because I am not taking pleasure in farmers' yield losses, but I'm just astounded by biology. And I'm also very appreciative of the biology I was taught in a small school in southwestern Pennsylvania. And I just see it playing out in front of me in the fields of Iowa every day.
All right, so if folks want to learn more about what the SCN Coalition is doing to help eradicate these pests, where do they find that information?
So it's all on an aptly named website called the SCNcoalition.com. There's no spaces, so SCNcoalition.com.
All right, Greg Tylka, thank you so much for the time.